In the biochemical characteristics lab, you're going to need a sample of bacteria. In this case, here we go. You can see that it says BC. That stands for Bacillus cereus. I showed you in the back of your lab manual where that Burgess is, the Burgess manual or table. The reason that I started using these abbreviations is because these plates are fairly small to write down Bacillus cereus across the bottom. So I just decided to use the abbreviations. So I have my sample. For the catalase test, I need a nutrient auger plate. So you can see that it's labeled NA217. I made it on the 17th of this month, and it's nutrient auger. I also needed to do a starch digestion test. So here is a starch plate. The starch is in the actual auger itself. And as I said in the previous video, in the lecture video, if you put starch and iodine together, it's going to turn black. If, however, you've had starch digestion with your sample, you are not going to have to worry about seeing black because there's no starch there. So the iodine stays amber. The sim tube. It's solid. It's not liquid. So here's the clean sim tube. And you can see that it's pretty see-through. It's pretty clean. It's pretty clear. This is what we use the inoculating needle with. We use it basically to stab into our sim with our bacteria, and then when we come back in 48 hours and look, we're going to see if the bacteria stayed in that stab line or if it actually moved outward. Instead of being clear like it is here, it'll look cloudy. It'll look very cloudy comparatively. We also have our phenol tubes with different types of sugar. So here's the Sucrose tube. It is red, and I've got that Durham tube, the upside down baby test tube in here that I talked about. I have the dextrose tube, and again you can see and hear that that little tube is in there. I've got the lactose tube, and you can see the little tube there. Now, this gets a little confusing. If you look at the top here, it almost looks like there's a bubble, but that's actually the glass at the base of the tube, just like the glass here. Keep that in mind as well. Then you have maltose. Now, the sad thing is this is supposed to be red, like glaringly red. And unfortunately, because our media got fairly old, it kind of changed and it's now more of a yellow color. But regardless of that, when we do our experiments, we're still going to be able to see a change between the control and the inoculated sample. With the way that we do this experiment, in lecture, with chapters 9 and 10, we talked about incineration. One of the things that I mentioned was that we do this in the lab with our inoculating tools because we put them directly into the fire. So because we are dipping these into a tube that is fairly long, when you incinerate anything that could be living on this, you always go all the way to the base. You don't just want to do the tip, because remember, you're actually poking this into something that is fairly deep. One of the things that you have to do when you're preparing to do this is label everything. I am Ronica Williams, and the bacteria that I am going to use in this example is Bacillus cereus. So I label my plates. There's the starch one, here's the nutrient auger, Ronica Williams, Bacillus cereus, sorry. And I label my tubes. If you're in a class of 24 and you've got 24 people doing the same tests as you are, you can't just leave it to chance that yours will be the only one without a label. Ronica Williams, RW, and then this is Bacillus cereus, so BC. Hope you can see that. There we go. And I'm going to do that with all my tubes. With the phenol tubes, it doesn't really matter if it's on the side of the tube because you're looking more for color than what the inside looks like as far as cloudiness. With the sim tube though, it's going to be a bit different. I'm going to take this label off. I want to be able to see clearly the sim and see through the sim media. So I don't want to have any writing or anything down here where that is. Instead, I'm going to write up here, above where that sim is. We've got these labeled, we've got these labeled, and we've got our sample. We're going to open our sample, take the parafilm off. When I work with bacteria, I'm going to scoot that out of frame a bit so I can put these more into frame. When I work with bacteria, I tend to work with it upside down. If you look, the bacteria are actually on this side of the plate. This is the lid. You'll notice that this is a nutrient auger plate, the date I made it, and the date I actually spread the bacteria. Now you could put that all down here and that's fine, but for the sake of me taking pictures, I didn't want too much busy underneath this plate. Also, so that you're aware of how fast I'm doing this, I have multiple inoculation loops that I have already sterilized in the fire. That way I can do this more quickly instead of taking up a lot of time. So this has already been flamed and it has been cooling in a rack with all these other inoculation loops. So they're not touching each other, they're not touching the counter, and I've already heated them. I'm going to take my sample. I'm going to get a little bit of bacteria on my loop. So if you look, see that little lump of bacteria right there? Now I'm going to get my open starch auger plate and I'm going to spread it around on the auger. Now the cool thing about this is I know that my sample is pure. I know that it is not contaminated with anything else because I made it fresh. So I don't need to do a streak plate because all I'm really interested in is seeing if this has starch digestion. I'm going to put this to the side since I'm done with it. The next excuse me, plate is the nutrient auger, the one that I'm going to do the catalase test with. So again, I've already sterilized this second loop. I'm going to grab a little bit of bacteria on my loop. Again, you can see the little lump. Flip my nutrient auger around and spread this on my nutrient auger. Doesn't have to be neat, it doesn't have to be clean. I'm done with that plate. Now moving on to our tubes. Here's my sucrose tube. Because I'm about to put this in the fire, I'm going to take this tape off. Now if you're wondering how I keep them straight, these are actually labeled. See the S right there? I just figured that it would be easier to see on the orange tape. So, Sterile Loop, I'm going to hold on with my pinky, unscrew the lid. Got to clean the mouth. I've got to sterilize the mouth of the tube, so I'm going to put it through the fire a couple of times. I'm going to grab my sample. Again, get a little bit of bacteria. Got my little bit of bacteria. You can see the lump. I'm going to put my inoculation loop in the fluid. And I'm going to swirl it around. Try and knock that bacteria off of the loop. Careful so that you don't fling it everywhere. Again, go through the fire a couple times. Close it up. And flame your loop. Don't know if you can hear it, but you can hear the sugar burning. The fire even looks brighter because it's burning the sucrose off of the loop. Now remember, I went into that tube pretty deep and swished it around. So I'm going to go from the tip all the way to the end to make sure that this is all clean. Dextrose. Take the tape off so that I don't burn it. Label. There's the D for dextrose. I'm going to go through the flame. Grab some bacteria. See the lump of bacteria? Put it in the fluid. Swish it around to get the bacteria off. Go through the flame. Cap it. Always in my pinky. Now, dextrose is on this now. That's another sugar. That flame sizzle you hear is because of the dextrose. Dextrose actually burns really well. And I go all the way to the end. Since I went deep into that tube, I want to make sure that I didn't get any bacteria all the way up here. Because if I were doing multiple samples, I would need to make sure that this is as clean as can possibly be. I don't want to mix my samples. Here's lactose. Last liquid media is the Maltose Tube. So just as a reminder, the reason that we are using 4 different sugars is because depending on which bacteria you have, it may or may not eat this particular sugar or ferment this type of sugar. So if it does, you get a change in the phenol color. Instead of being red, it turns yellow. Get in sample. If it doesn't, it will stay this original color. Now for me personally, especially with the maltose, because the maltose is already so yellowed, I actually keep a control sample so that I can do a comparison between what this looks like once the bacteria has grown and what the original color was. Here's my sim tube. Sim is solid, it's not liquid, and the test tube you use is actually a flat bottom tube. Because I'm going to have to flame this, I'm going to take this tape off, but I wanted you to see what it was. I'm going to label it RW, actually, there we go, RW, BC, for Bacillus cereus. You're going to stab in the middle of your tube with your needle. When you do that, that wound that you created in the gel will be visible when your bacteria grow. If your bacteria do not grow, if they just stay in the stab that you make, you'll see a line dead center in the middle of your sim auger, and that's all you'll see. However, if they actually move into the media around where you stab, this is not going to be as clear as what you see here. See how you can see my fingers through it? The bacteria will actually start moving into the auger on the sides and spread. And as that happens, the sim media gets very cloudy. I'll show you a picture of this right here. I've already sterilized this, so I'm going to get my sample. I don't need much, so I'm going to take my needle, just going to rub up on the bacteria here, just a little bit, to get just a little tad bit of bacteria on the tip. This is still a tube, so I have to flame it. So I'm going to flame the mouth of the tube. Now, the goal is to get it dead center, straight in, straight out. Sometimes it works that way, sometimes it doesn't. Just kind of depends. So I'm going to try to stab into it and then come straight back out. Flame the mouth of the tube and close it up. You're going to flame and cool your needle, again all the way to the end, because you actually went into your tube. Right now, you can see see that little line right there? You can see where I stabbed it. You can also see that it's not necessarily the straightest stab ever. But when the Bacillus cereus grows, we're going to see if the Bacillus cereus is modal, if it's moving, because it will be beyond where we stabbed. If it doesn't, it'll stay right there in that stab wound that we've created. We have our phenol tubes. We're looking for acid production, which means that it ate the sugar that's in there, specifically, whether it's sucrose, dextrose, lactose, or maltose, and produced acid. And with the durum tube that's in there, see how there's no air in there? We're going to see if there's any air after it's incubated for 48 hours. This is the biochemical, well, along with these two, are the biochemical characteristics experiments that you do. For the plates, we always seal them with paraffin before we put them in the incubator. So I'm going to seal this with paraffin, seal this with paraffin. Now we can put them in the incubator. So here is one incubator. This is the 37 incubator. Hope I don't make you sick. This is our other incubator. This is the 25-degree incubator. As I showed you in the Berge's manual, different bacteria like different temperatures. Since we have 2 incubators, I know that Bacillus cereus likes 37. So when I put this into the incubator to incubate for 48 hours, I'm going to use the 37-degree incubator. Open it up, put my plates in. And as you can see, we've got other plates from my students. My face-to-face students, I should say. And as you can see, my face-to-face students have their own plates in there incubating. I had my sim tube and my phenol tubes, sucrose and dextrose. You can see the S and the D, lactose and maltose. Now for reference, I also made a Micrococcus luteus tube because this one, again, from my Berge's manual, I know that this one is a 25-degree bacteria. It likes it at 25 degrees. So in this case, I'm going to put it in the 25-degree incubator.